Any other general questions? Okay. Yes. Yeah, just remind me. Yeah, I've got them for each. Yeah, I'll put up more sample questions. I just need a little prompting. Okay. So really, what we're doing, we're going to finish up chapter 8 today, and we should be able to start into chapter 9. Two very different areas. So what we're doing in this chapter is we're looking at rather large morphological changes in chromosomes. So you got to sort of think larger scale for this chapter. So we're looking at differences in chromosome structures that you can actually see using the light microscope. So if you can see these abnormalities in the chromosomes, you're dealing with clearly more than one gene, and depending on the organism, who knows how many different genes. So it's just, again, we're going sort of on a bigger scale. Then as we go into chapter 9 is the one chapter we'll deal with on bacterial genetics, and so we have to think a little bit differently on that one. But let's just take them one step at a time. So let's start out with our two questions that we've been doing. I'd play around with the lights in here, but I'd probably put us in total darkness. But see if you recognize that image from the last lecture. Yeah, looks like most of you do. So we'll just go to 1 minute on this one, let everybody get settled in. Okay, 3, 2, all right. So this is obviously what? This is the fluorescence in Situ hybridization. So this is a very widely used technique. In this case, the chromosomes are prepared under conditions where you can still see the morphology of the chromosomes. And then the probe, which is kind of hard to see under this lighting, this little spot over here and this little spot over here is tagged or labeled with a fluorescent tag so that we can visualize where that probe is localized. And in this case, we know that that particular probe, which is a DNA sequence, it's part of chromosome number 9. Okay. And a straightforward question about something we'll be talking a little bit later today. And 1 minute should be good on this one too. All right, what's this going to be? So these are aneuploidies. We'll talk about this a little bit later. This is either the gain or loss of a specific chromosome. Okay, so we're continuing in this chapter 8 on chromosome variation. So last time we talked about duplications. Let's see, what did we do? We did duplications. That's probably about all we did. Okay, so we really just did the duplication. Didn't get as far as that. Okay, so now what we're going to do is the opposite of that. Instead of having duplication, so we remember that was an example that we had was the Bari mutation in Drosophila, where we have multiple copies of this DNA sequence that's on the X chromosome, and the more copies that you have, the more extreme the phenotype. Here's a deletion, which is exactly what its name implies. Some sequences have been deleted or removed from the chromosome. So in this case, this segment, which is just arbitrarily identified as E through F, is now missing on this chromosome. Would this be lethal? Well, could be. But with the heterozygote, it's usually not a problem if this isn't a huge deletion. Because why? Are there still at least one copy of the genes present? Okay, so there's at least one copy of the genes here. So usually a heterozygote with a normal chromosome paired up with this deletion is okay. However, the outcome of these deletions and what happens really depends on where the breaks occur and how much is lost. So that's what I was saying here. A couple of extreme cases. One is if the deletion were to remove the centromere. Would that be a problem? Yes. When that happens, then usually the chromosome can't segregate properly during meiosis and mitosis, and then that usually ends up being lethal. Here's a very important point, and it'll play into one or two questions on the exam. Almost uniformly, it's been found that homozygous deletions are lethal. So what does that tell you? What does that tell you about the DNA sequences that have been lost? Right. So what's been found just empirically is almost any size deletion that you can see and really know it's there under the microscope contains at least one essential gene that's required for proper development. And so when those deletions are homozygous, they're almost invariably lethal. Okay? All right. Okay, so there's two things we got to talk about here. So have we already? Okay, no, we actually did talk about a deletion mutant, right? Okay, so this is what we were talking about before. In that, if the deletion removes a portion of the chromosome that has the gene you're interested in, and the other chromosome has the recessive mutation, then what phenotype do you see? The recessive mutation. So in the jargon, the deletion reveals where that mutation occurs. And so we use this term pseudo-dominance. It just appears to act as a dominant mutation in the sense that it's revealing where this other gene is located. Okay. Now, here's where it gets maybe a little counterintuitive, so follow with me on this. A deletion would be a loss of function mutation. That means you're losing the gene. You don't have the functionality of that gene. Loss of function mutations are nearly always recessive. However, sometimes these loss of function mutations can appear to be a dominant mutation and actually act as a dominant mutation if they're what we call haploinsufficient. Everything, so far, all the situations we've talked about is we said, okay, as long as you have one functional copy of the wild type gene, then everything's fine. It can make enough of the gene product. In some cases, that's not good enough. In some cases, one fully functional copy of the gene does not produce enough gene product, and therefore this loss of function mutation really acts as a dominant mutation. That is, having that allele, that deletion, is sufficient to give you this mutant phenotype. So let me give you one example.
Is that what dosage comes
No. How would you?
like when you were saying the genes that doesn't get like
But that's under a normal genetically controlled system. Let me go through this, see if this makes sense. So here's a phenotype that's called notch. I'll show you a picture of it in a moment. It's the result of a small deletion on the X chromosome. So females that are heterozygous, in other words, they have a wild type X chromosome, then they have this small deletion, still show this notch phenotype. They don't have a wild type wing shape. And that's because it's a notch is a haplo-insufficient gene. The one copy of the notch gene cannot make enough gene product to give you the normal wild type. All right. Now this is actually, notch actually encodes a cell surface receptor, so that males who have this small deletion on the X, those are lethal, because it's required. This is an essential cell surface receptor protein required very early in development. So only the females will have that notch phenotype. And if the female is homozygous for the deletion, then it's a lethal. Okay? So notch is haploinsufficient. So it is a loss of function mutation that is actually acting as a dominant mutation rather than a recessive mutation. But when it's homozygous, it's a recessive lethal. So, well, this is on the X chromosome, so the males aren't going to be. So males who have the deletion are just simply not. You're not going to find males that have the notch wings, because that's a lethal mutation. So what it does is think about what the gene product is doing. It's a gene product, a protein that's required and essential for development. You don't have this protein, the embryo never develops, and therefore it's lethal. It would be a normally recessive phenotype if one copy of the notch gene was enough to give you a normal wing phenotype. But it's haploinsufficient. Haploid, meaning one copy is not sufficient. So while it's a loss of function mutation, which would normally be recessive, it acts as a dominant mutation. Is that confusing enough? Good. I said it's going to be counterintuitive. This is where we're trying to take these definitions that Mendel gave us and fit them into much more, put them into genetic context where those definitions don't fit exactly. Yes?
Would it be possible for like a mutation or deletion to occur on like the next chromosome, but like say for instance like a female has another X chromosome, the one with the mutation becomes like bar body, and there's only like one X chromosome that's like becoming.
Well, you wouldn't have that in the flies,
Just, like, in general.
but
Would that be a possible thing? So like the deletion occurred, but you wouldn't see it.
well, if I'm trying to think about this, if I'm following the question, if you had a heterozygote female who was in a mammal, then you'd have situations where some of the tissues are expressing the normal gene, and that would be fine, and others where it'd be the deletion, it'd be the X chromosome with the deletion, and they wouldn't be expressing it. So depending on how that selection came about, I don't know what the outcome of that would be. Okay. So why? Yes.
Just to clarify the terminology of haploinsufficiency, does that only apply to deletions and other kinds of mutations or gene sequences?
Haploinsufficient would apply in any situation where having only one fully functional copy doesn't provide enough gene product to give you a wild type phenotype. So there aren't that many of them. In most cases, that's why most mutations are recessive. Most mutations are loss of functions. But as long as you have one functioning allele, then you just see a normal wild type phenotype. Okay. So while you're trying to figure that one out, why don't we try this question? Let's see. What happened? What?
On the right side.
The right side of the questions are
Oh, I wonder why they did that. All right, let's just take a shot at it. Sorry, it's some sort of a formatting issue I had with it. Yes. Where are we at?
Oh, never mind. Okay.
Yeah, no, it's just I got a formatting problem. So basically, if the centromere is deleted, then the whole chromosome could be lost during cell division. Recessive mutations may be expressed. That doesn't make much sense. So where I was going with, I'll just give you credit on that one. Here's what I was trying to go with this was these haploinsufficient genes can result because too much gene product is made. That's not what's going to happen. But the software is fighting. Yeah. Okay. Well, that wasn't very productive. All right. Another situation, and it'll get a little bit complicated and some funny drawings, but let me see if I can walk you through this. So we've already looked at duplications, we've had deletions. What we can look at are now inversions. Now we're going to make an assumption that no genetic information is being lost, but rather we're just going to break the chromosome in two places, so you really have three pieces. One piece is going to flip over, and so its position is inverted relative to where it was, but all the genetic information is still there. And then ask, what's the consequences of this? And to make this, of course, to make this complicated, we have paracentric and pericentric inversions. All this means is if the break occurs on opposite sides of the centromere, if the centromere is involved, the centromere is part of the inversion, then it's a pericentric inversion. If it's anywhere else and doesn't involve the centromere, then it's paracentric. Otherwise, everything else will behave exactly the same. Okay? That makes sense? So with a pericentric, you have a break on each side of the centromere so that when that piece flips over, the centromere is part of the DNA that got inverted. Okay? So we're going to assume no loss of genetic information. We're just changing the order of the genes on the chromosome. Okay, so that's in a very simple cartoon. That's what they're trying to show right here. All right. You would think, yes?
What happens when a pericentric inversion happens and when the homologous chromosomes line up during...
We're going there.
Oh, okay.
That's where the funny drawings come in.
Okay.
That'll be the part I'll have to walk you through. Okay. In most cases, you would think, well, that shouldn't affect gene expression. It does and it doesn't. Depends on when it happens. You can have what are so-called position effects. We'll get into this more when we get into the structure of chromatin, but there are regions of the chromosome which are in a very tight, condensed condition because basically there's no transcriptional activity going on. There's no RNA being produced. It's a very quiescent. So that could be over here. This gene needs to be actively expressed, but when you inverted it, you brought it into a part of the chromosome that's now quiescent. So all the position effects means is how that gene is being expressed is being altered because it's being put into a new neighborhood, if you want to think about it that way. Yeah.
Would you kind of call a position effect like an epigenetic phenomenon? Or is it not because it's not like methylation and stuff like that? It's just more like...
That's a good point. I mean, technically, epigenetics is anything that affects gene expression without changing the DNA sequence. Well, you're changing the DNA sequence, but you're not changing the DNA sequence. So that's an interesting point. And I'm not sure which. I think you could probably argue it. If it was like one of those essay questions, it would be how you phrased your argument. Okay. So the main thing we're going to be looking at is the very question that was raised down here. What really happens during meiosis and crossing over? So we're not going to really worry about genetic effects, gene expression. We'll assume that's not much of an issue. Also, if the inversions are homozygous, that's not a problem for genetic recombination. Remember, the chromosomes are going to line up nucleotide by nucleotide along their length. But if they're both identical because they're homozygous for this inversion, it's not a problem. The problem happens with the heterozygote. So it's going to be pairing a wild-type chromosome with the chromosome that has the inversion. And that's where you get into these kind of crazy drawings. And in this particular case, this is from the textbook. I put them in there. But I think this figure that I took from another textbook, I think is clearer. So you can either use the figures in the textbook, or you can use this one. Whichever one helps you visualize it a little bit better. So here's the problem. I wish that was a little clearer. Okay. The chromosomes have to line up sequence by sequence. This outer chromosome, this blue chromosome, number 1, and number 4 down here is not a problem. It's when you have the wild-type sequence that now has to pair with the inverted sequence, which gives you this complicated looping structure. So all that's going to happen out of this, and the best thing to do is look at these two chromosomes and just trace them out. So, okay, so this is a paracentric. So the centromere is over here, and the inversion is actually over here. So the inversion's in the middle. So if you have crossing over, because they're all paired up by their sequences, you have big A, big B, big C, and it crosses down to here, okay? And then if you follow it around, it's got small d, I think it's small, and then it comes back out over here at A. It ends with number 4. So that's this chromosome. And in the opposite case, you have 3, so you got small a comes up through here. Let's see. Try and control. Oh, I was going to say that doesn't make sense. Number 4 down here. So you got this comes up to D, crosses back up, comes out, and number 2 over here. Get the figure, look at it. Here's the point. Let's see if I got a better figure. Yeah, so here's the point. When you trace through there, what you're going to see is some of those DNA sequences are duplicated and some are deleted. All right? Also, this chromosome has 2 centromeres. So when you have the paracentric inversion, you're going to end up with one chromosome that has the 2 centromeres. Is that going to be good? No, that's probably going to get lost. And then the other chromosome has no centromeres. So what this means is any crossovers that occurs within the inverted region is going to result in non-viable gametes. So in the jargon term is sometimes used that these inversions suppress crossing over. Do they prevent crossing over? Do they prevent recombination? No, they just lead to non-viable outcomes. Now this is just if the recombination occurs within the inverted region. If you had a recombination that occurs out over in this region, is there a problem? No, it's only in that part where it's all looped around and paired up. All right. Now the pericentric, you get exactly the same outcome. Again, I find these figures a little confusing myself. The same sort of thing. The only difference is the centromeres are now part of the inversion. So when you go through each of these, and the 2 that I've marked with the 2 arrows are the 2 recombinant chromosomes, do any of those have 2 centromeres now? No, they each have 1, but they still have duplicated and deleted regions. So for instance, let's see if I can read here. I'm sorry, I just switched it, came out a little better. Here's a clear example. All right, so you got A, I think A here. So you got 2 copies of A, but D is missing. So the take-home message is any crossovers between the wild-type chromosome and the inversion will lead to non-viable gametes because of a loss and a duplicate. Some sequences will be deleted and some sequences will be gained. Okay? So you don't recover those. Those gametes are non-viable. All right. So now we got 2 really confusing things. Okay? I think if you take the figures, trace through them, and look at the outcomes, you'll see it's that. So the whole important point is it's just the crossing over that occurs between the wild-type chromosome and the inverted region. Crossing over anywhere else on those chromosomes is fine. That's really the only major outcome of these inversions. Okay. Anybody understand that well enough to even ask a question? Okay, it's going to take you guys. Yes. Mm-hmm. That's the centromere. So this chromosome right here starts over here at 2'. So there's big E. Okay, comes along here. Goes up to big D. Up here. Crosses over. The small C. Then B comes back around. Here's B. And it comes back out here at 4'. And now you have E again. So what happens is you have 2 segments with, you got 2 copies of E, but you have no A. So it's duplicated some DNA sequences and it's deleted some A sequences. So this chromosome and this chromosome, they're going to end up in gametes, sperm, egg, whatever. They're going to be non-viable. They're missing DNA sequences and they have duplicated sequences. And so those will be non-functional. So these recombinants, you never see them because these gametes are non-functional, non-viable.
What exactly are we looking at with the loop here? What is that representing?
So the reason it's looping around, and I could, if I had better artistic abilities, I could probably draw it, is the sequences are inverted, but the chromosomes have to line up by their DNA sequences.
Okay, so they're lining up the sequences and.
Right, so basically the inverted sequence is having to loop back so that they're all aligned. See, so you still have segments A, B, C, D, and E aligned with each other, but part of it's been flipped over. The only way for that to happen is for this, at least the way the artists draw it for us, is for them to reverse and come back.
So how did it become inverted in the first place?
Through a chromosomal aberration, in other words, you can have, we'll talk about this later on when we do like DNA damage, but you can have X-ray, some Drosophila geneticists did this on purpose. You put flies in an X-ray chamber, you hit them with X-rays, the X-rays break the chromosome. So the question was, how did the inversion happen in the first place? Some event, usually high energy cosmic radiation or something, really just shears right through the chromosome. Literally just cuts the DNA like you took a pair of scissors. These broken ends like this are very unstable in the cell. And the cells don't really know how to pair them up. And they just start knitting them back together trying to repair the damage. Well, if that one segment got flipped over, and this piece joined here and this piece joined here, you now have an inversion. So it's the result of DNA damage. And this is the consequence of it. Yes?
So for something to be functional, it must have all the letters.
Having too much DNA or too little creates a problem. So it needs to have the right amount. So each of these recombinations within that inversion ends up creating too many copies of some DNA sequences and completely losing others.
Does it matter if it's dominant or recessive?
it's so much. It's such a large loss. If the cell doesn't, it's such an imbalance of the genetic information. The cells just they can't deal with it, and so it just becomes non-viable. You got a question? Yes.
So on the application side, would that be a reason why radioactive rates or radioactive rates are bad for 80% of the
Well, high energy radiation. Yeah. We'll talk about this a little bit later on, but high energy radiation, like you get from accidental exposure to nuclear reactors or some sort of occupational exposure, or if you happen to be in the vicinity of an atomic bomb, something like that. Yeah, you can create so much damage that it's pretty much lethal almost within a very short period of time. One other question is, what about the small amount of radiation that we all get as we walk outside and there's cosmic radiation? But as we'll talk about, cells have very efficient mechanisms for repairing DNA. DNA damage is happening all the time. We're all losing a lot of purines as you're sitting around listening to me here. But hopefully your cells repair this. So it's actually a very efficient process. But if you exceed the capacity of that repair system, you're going to end up with damage. You're going to end up with something like this. Yes?
So in the resulting damage, only 1 and 3 would be viable?
Yes. That's what's going to happen is the second one and the fourth one, let's say those are in sperm, they would end up, they could fertilize an egg, but the embryo would never develop.
So like how is the first example just like this
Well, the questions would probably revolve around, under which situations would you end up with non-viable, I could show you the crossing over and ask, okay, would you get, if you had a crossover, in other words, if the crossover occurs anywhere between this little corner and here, you have two non-viable gametes come out. If it occurs outside that zone, then you're okay. Or if they're homozygous chromosomes for the inversion, you're okay. All right, so let's, at least we got it all on the screen. Let's see what happens here. Yeah, 1 minute should be good on this one. Does it make sense? Excuse me. Okay. 3, 2, 1. All right. Okay. Well, that's like why I like to put up these questions and why it's challenging to write the exam questions. Most likely an inversion. Could a tandem do it? But the other ones could. Again, it's how you know. That's part of the process is that there aren't nice, clean answers to these situations. All of those, yes, could, in fact. You could argue under the right conditions. Inversion, most likely to cause it. Again, it just depends on where it happens and how much of the DNA is involved.
So if you chose D, do you get half credit?
Half credit? Let me see how I'm feeling when I go down the list. Yeah. I'll probably be feeling generous when I set the points on it. This is all part of a learning process. All right. Let's try this one. What do I got here? During meiosis, when crossing over occurs in a paracentric inversion loop, what percentage of the meiotic products result in non-viable gametes? Well, that's what we were just talking about. So, and this time I'm going to have to change it. Here we go. And there's the answer. So what the heck? Hang on, we haven't even started yet. There you go. You know, sometimes when the karma starts to come after you, it gets you. Come on, you can do this. Where's that 5? I gotta get that 5. All right. Well, I'll give you 45 seconds. Hurry up, figure out how to scroll down to get to the 5. See, this is just payback for the last question. All right. Okay. Since we, yes. The what? I give credit for both. It displays all the entries. So 0.5, 0.5, 50%. I give credit for all of that. Yes.
So in like an easier term to like recognize it, I guess, could you say that all the times that it accommodates is you just cut it out for that question?
For this
'Cause,
question,
like, based on that example, does the basically say that every time there's recombinant DNA in that sequence, that it's just it's deleted again?
the key part of this is it's got to be between the wild type sequence and the actual sequences that were inverted. If it occurs outside the points where it was inverted, then everything's fine. It's just that they're misaligned, or they're because of the way they have to realign through the inversion is why you end up with the recombinant chromosomes being non-viable. Okay. Yes.
For this question, would it matter if it was pair-eccentric without changing it?
No. All the para or para-eccentric changes is whether or not you end up with recombinant chromosomes that, whether you end up with recombinant chromosomes that only have one centromere, but it's still non-viable because there's been a gain and loss of genetic information, or whether one has 2 centromeres and the other has none, and that's enough to make it non-viable. Okay. So I think this is the last rearrangement. Don't confuse what I'm going to tell you about here with what we just did. The outcome of this is going to be that 50% of the gametes, I'll just jump to the end of the chapter, not the end of the chapter, the end of the story. 50% of these gametes are going to be non-viable. The one we just did said that only when the recombination occurs within that inverted region do you end up with 50% of the gametes coming from that meiosis being non-viable. So make sure you see the difference between the two things I'm going to talk about here.
So this one's 50% non-viable regardless.
Right.
Like all the
This is what's going to happen here is you're going to be fine, but what you actually observe in the lab is that the fertility appears to be lower. You're not getting as many offspring as you expected, and that's because half of the gametes are going to be non-viable. So these are translocations. Here we're talking about, everything we've talked about so far has been rearrangements of DNA sequences on the same chromosome. Translocations are actually taking DNA from one chromosome and exchanging it with a non-homologous chromosome. I'll show you an example with chromosome 21 and chromosome 14 and such. So we're talking about exchanges between non-homologous chromosomes. A non-reciprocal translocation means just a one-way transfer. One part of the chromosome goes to another one. That's fairly rare, and we're not going to talk about any examples of that. The most common is a reciprocal translocation. You still have all the genetic information. It's just portions of one chromosome have now exchanged sequences with another one. Okay. So what you're really doing, if you think about it, is you're putting the genes into different linkage groups. So genes that used to be on chromosome 1 might end up being on chromosome 2, and now they're linked with all the genes that are on chromosome 2. All right. The other is you can affect how the gene is expressed depending on where that chromosome breaks. All right. So for instance, later on, when we get to the section on cancer genetics, hopefully we get to that, what you find is certain types of cancers, all those patients have the same translocation. And what's happening is basically a oncogene, a gene involved in cancer, the chromosome is breaking This gene's getting transferred to another chromosome when it comes under control of another promoter, and basically it is expressed at the wrong time, in the wrong place, and in the wrong amounts, and leads to cancer. So you can affect how a gene is expressed if the chromosome happens to break at a critical point. Okay. The other one I'm going to talk a little bit about, and I'll show you a form of inheritable or familiar Down syndrome, is a Robertsonian translocation. Here you're going to take two acrocentric chromosomes. Acrocentric means the centromere is where, relatively speaking? Fairly close to the end. So you're going to take two acrocentric chromosomes and join them together and make one metacentric chromosome. So you're going to take two chromosomes and make them into one. I'll show you an example a little bit later. Now, if you think through on that one, it's like, wait a minute, something's getting lost. It's not going to work all the time. So it's got to be acrocentric so that the little short pieces that are going to get lost aren't going to be viable, they're going to have to contain sequences that aren't essential. And one of those centromeres is going to have to become inactivated. So the right set of conditions have to happen, but selected through enough time, it does happen. I'll show you a few examples. Okay, so that's what's on there. All right, so that's really what the Robertsonian is really just trying to show you here is we have an acrocentric, so here's the chromosome. We have a break, these two chromosomes. These fuse together to now create a metacentric chromosome, but it's got to have one functional centromere, can't have two, and the small little fragments that come out of here get lost, and as long as they're not essential, then the system could remain viable. So it's not going to work every time, but I'll show you a couple of specific examples. Okay. All right, back to chimpanzees. So here's an example. We talked about this before that when you sequence the genome of chimpanzees, you compare it to humans, they're about 95, 96% homologous. If you look at the pairs of chromosomes, okay, humans have 23 pairs of chromosomes. Is that right? 23? But the large primates have 24. What it looks like is there's been a Robertsonian translocation, whereas two acrocentric chromosomes in primates have now formed a metacentric chromosome in humans, which would be chromosome 2. So if you actually line these chromosomes up and look at the banding patterns on them, so here's the acrocentric chromosome from chimpanzees, gorilla, orangutans. It all lines up very nicely with chromosome 2 in humans, and now you got direct DNA sequence data that shows that it really looks like these two acrocentric chromosomes came together and formed one new metacentric chromosome. Yes?
Okay.
Oh,
Does that reverse aging?
is it reverse aging?
Yeah, because different the telomeres, when they get small, different that
We're not,
end of their age.
yeah, we're not, the telomeres are on the ends down here.
But if you connect two, no?
No, it wouldn't affect it. Any, was another question? Yes.
So, does Robertsonian translocations increase the number of chromosomes by half? Is that what it is?
No, in this case, it reduced it by one pair.
Oh, yeah.
We had 24 pairs in the primates and 23 pairs, because two that were in the primates are now one. Okay. Here's the main thing I want to show you, and again, I'm going to use a figure from another textbook. Hopefully it's going to come up a little clearer. We got to watch this sort of funny pairing of the chromosomes. So it's the same rules. The chromosomes have to pair through meiosis based on their homology, regardless of whether that sequence happens to be associated with some other non-homologous chromosome. So what we really want to do is look at the outcome of meiosis when you're dealing with these translocations. Okay. So in this case, here's a translocation. So we have this, here's the wild type chromosome. All right. So, and over here is a non-homologous chromosome. And what we've really done is exchange the sequences that are part of this chromosome. I guess this is brown. Is that right? So this sequence is now over here. So it broke right here, and now it exchanged between this blue chromosome and the brown chromosome. Okay? So you see how it's a translocation? There's sequences between non-homologous chromosomes. They get paired up sequence by sequence. And here's the key figure. There's two ways that the chromosomes can line up at that metaphase plate during meiosis. If you line up so that we just arbitrarily call 1 and 2, chromosomes 1 and 2 are the blue homologs, 3 and 4 are the brown homologs. If they pair up this way, so that when they segregate and you get chromosomes 1 and 4, over here you get the two full normal chromosomes, and as the translocations there are gone. It's gone. Over here you get the 2 translocated chromosomes, but it's called a balanced translocation because it still has one copy of each of the genetic elements. There's no extra DNA, there's no gain or loss of DNA. So these are viable. 50% of the time, they can line up like this. So now all we've done is we've kept 3 and 4 in the same position, but reversed 1 and 2. Now you get one normal chromosome and one translocated chromosome. And the same thing over here. Now what's happened? You have duplicated sequences for D, and K is missing. And over here you have duplicated sequences for K, and D should be missing. Are these going to be viable? No. So that's why with the translocations, half of the gametes are going to be non-viable. And it just has to do with the segregation of the chromosomes through meiosis. Make sense? Okay. All right, so let's try this question. Oh, I got to go back. All right. Here we go. Okay, 1 minute should be enough on this one. Yes. Would be different than.
For example, half of the cells are viable.
Oh, those are just the gametes. So that just means that, let's say that was a male and he was sperm, only half of the sperm will be actually, half of the sperm that fertilize eggs will not lead to won't have a viable outcome. The gametes would affect it. So you're not gonna have any progeny. So what you see is the progeny that are born are fine. It's just it looks like a reduced fertility. You're not getting as many as you normally would because half of the gametes, half of the eggs, or half of the sperm are not viable.
So assuming this and inversion have the same outcome, like even though you have to tell them apart?
No, because here that's the important point. Here, half of the gametes are going to be non-viable. With that inversion, the important point is only the ones that are only the recombinants that involve that occur within the inverted region lead to non-viable gamete. If that inversion was very small and recombination didn't occur very often, you may never even know this. Yes?
This is the translocation, the reason why the SRY gene can end up on the pseudo-auxotrophs of the X chromosome.
No, that'd be a real problem. But they did found some cases where the SRY gene actually got onto an autosome.
Oh, okay.
And so while it was an XX karyotype developed as a male, and then they tracked down and they found it was a small translocation onto an autosome. Okay. What? I told you, karma's a bitch. All right, so what's the question? What's the question? But those are non-viable.
But it doesn't ask if it's viable or not.
Oh, it doesn't. No, it doesn't cause. Okay, good point. It doesn't cause duplications on the chromosome. Okay, this is where you get into the minute. It doesn't cause duplications of the sequences on the chromosome, but because of the pairing of the chromosomes, then you can end up with the duplication. Because of, I'm sorry, the segregation of the chromosomes is how you end up with the duplications. Right. The chromosomes themselves do not. Look on the individual chromosomes; they do not have duplicated sequences.
So after translocation, you can't.
No, with the translocated chromosomes, they don't have duplicated DNA sequences. But depending on which homologs they pair with, because they involve different homologs, you can end up with a gamete that has duplications. Okay. I see your point in terms of the limitations of a one-sentence answer to a multiple choice question, to complicated problems with, and trying to answer a complicated question with one sentence. Yes.
Would they really be duplications if it's happening between non-homologous chromosomes?
You just end up with duplications in the gamete.
Yeah.
But the actual chromosome doesn't have the duplication, which is where I was trying to go with that question. Okay, since I'm having such a good day, this should be a little bit easier. So the last part of this is just aneuploidies and polyploidies. The aneuploidies will be the main thing we're talking about. With aneuploids, all we really mean is a gain or loss of one or more chromosomes. And in most cases, except for the sex chromosomes, these are going to be non-viable situations, especially in animal cells. They just don't tolerate extra DNA sequences or a loss of DNA sequences. Okay. So just some of the terminology. Nullisomy just means the loss of both homologous chromosomes. So null or zero. Monosomy would be a loss of a single chromosome. Trisomy, the one you're probably most familiar with because for the Down syndrome would be, and this is what that first question was, would be a gain of one chromosome. So in this case, it would be 3 copies of chromosome 21. And you can go to tetrasomy and you can have higher and higher levels. Again, in most animals, these aneuploidies are not tolerated if they're on the autosomes. They are a little more tolerated with the sex chromosomes because of things like the Barr body where you can begin to balance out the gene dosage. Okay. And so that's really what this is. And in most cases, because of these gene dosage questions, these type of aneuploidies are usually lethal, except for the sex chromosomes as we talked about before. So these become the most, so the sex chromosome aneuploidies are the most common that we see in humans. We talked a lot about that back in chapter 4. Here just reviews it. I guess there's some data that says, for reasons that probably aren't clear, aneuploidy in human embryos is actually quite high, to the point that at least some studies suggest that maybe almost as many as 30% of all conceptions spontaneously abort due to aneuploidies. Again, how do we get aneuploidies? Things like, remember, non-disjunction, these type of things. Sex chromosome, we've talked about those before. This is the one we can talk about a little bit, and also we'll talk a little bit about the Robertsonian translocation. And this is probably the most well-known aneuploidy, would be the one causing Down syndrome, which is a triploid for chromosome 21. The chromosomes were initially numbered based on their size, number 1 being the largest, number 22 the smallest, based on looking at these chromosomes under the microscope. Now that we've done DNA sequencing, it's realized that number 21 is actually smaller than 22. So 21 is the smallest. It only carries about 300 genes, and so this is probably why it can be tolerated. Now, one of the things we still don't quite understand is the degree of severity with Down syndrome isn't easily predicted based on this aneuploidy. So in some situations, the clinical symptoms can be very mild. Sometimes it can be very profound. They believe that the most likely cause is non-disjunction through the eggs. We've talked about that before. Okay. This is one I'll show you where this is a situation in which you can have it. So, as it says here, a couple who have a Down syndrome child, their probability of having another child with the same syndrome is only slightly higher than normal. However, because of a Robertsonian translocation, I'll show you one example of a basically inheritable form of Down syndrome, or what would be familial Down syndrome. So it's a translocation between chromosomes 21 and 14. Since both of these are acrocentric, it creates a new chromosome and exchanges these sequences. So again, we're going to involve 21 and 14. Okay, we kind of go through this. So here would be the normal parent and the normal gametes. What you have here is a parent who's a translocation carrier. So instead of having 4 chromosomes, you should have 4 up here, right? 2 copies of 21 and 2 copies of 14. Here's the Robertsonian. So they have all the DNA sequences. So this individual's fine, but different things can happen during gametogenesis or through meiosis. So you can end up with a case where you have just the single fused chromosome, or you can end up with 1 copy of 21 and 14. You can have the Robertsonian chromosome in 21, or you can have just 14. Okay, and you can have these possibilities. So you can trace this through, and what you find is here you're back, assuming a normal gametes from over here, you're back with a translocation carrier. So this person is okay, but they still carry the Robertsonian chromosome. This one has now lost the Robertsonian chromosome. Why does this one have now, why does this child have Down syndrome? Because he's got the 2 normal 21s plus the Robertsonian chromosome, which gives it 3 copies of chromosome 21. Here you have a loss of chromosome 21, an aneuploidy. So this will spontaneously abort. All of these situations will lead to spontaneous abortion. Okay. So this is just one form of an inheritable form of Down syndrome, the result of a Robertsonian translocation. Okay. Any questions? All right. And continuing with the theme of this chapter, something else that doesn't make sense. Uniparental disomy. All right. So you have a let's say, so you have a couple, they want to have a child, and one of the individuals had a sibling with Down syndrome, with, let's say, cystic fibrosis. So there's a concern that, well, maybe that person's a carrier, and if their partner's a carrier, then there's a 25% chance the child will have the disease. They do a DNA analysis, and everything's fine. The one partner has 2 wild-type copies, and the other one is a heterozygous carrier. So everything seems to be fine. And then once in a while, it's very rare, they end up with a child who has cystic fibrosis. So the question is, it's a fully recessive autosomal disease, so how did they end up with having 2 copies of this recessive allele when the DNA analysis had confirmed that only one parent was a carrier? When you look more carefully, what you see is it's actually 2 copies of the same mutation. It's actually 2 copies of the same homolog from one parent. All right. Would that happen normally? No. Okay. How it happens isn't really clear. But what is thought that it probably involves an aneuploidy for chromosome 7 and then a subsequent loss of one of those. So you end up, instead of having this triploid copy of chromosome 7, which would lead to a spontaneous abortion, you end up with 2 copies of the one chromosome, 1 copy from the other parent, then you lose one, and now you end up with a duplication of the homolog from the one parent. It's not a completely satisfying explanation. There's still some of the science is unknown, but it does happen at a small but measurable percentage of times. Okay. And to kind of wrap this up, polyploids, generally you think of these mostly for plants. At least I've never heard a good explanation for why, but plants seem to like having extra copies of, I'm sorry, this is polyploid. We're talking about full sets of chromosomes. So instead of being diploid, which most animals are, these plants can be triploid or tetraploid, even higher. So these are full sets of chromosomes. And in many cases, plants that are polyploid end up being more vigorous, stronger, larger. Does anybody know why? I'm just asking because I'd like to know. I've not had, I don't know. Should I say? I'll tell a story. You guys probably haven't heard about, there's this thing called marijuana. You've probably heard about it before. It was only around when I was in college. And they, because this was before the internet, you had to get all your mail order stuff through magazines and this sort of thing. And I was just surprised there were these, High Times and some of these other magazines were selling inhibitors of meiosis, basically drugs that interfere with the spindle apparatus, which you could use to treat with plants to make them polyploid, because then that's one way to get a plant that now the chromosomes didn't segregate properly through meiosis, and now you end up with a diploid cell, and then you can make, so the idea was to make your polyploid pot plants, which supposedly would be better and such, but I guess what's going on in Colorado and else has become a real science. What you want? What the pharmaceutical companies making this stuff, that's what it's going for. Okay. So a lot of times, though, does it make sense? What you find is a lot of triploids, like triploid plants. I think bananas, are triploids. They're sterile. Does it make sense why plants that have an odd number of sets of chromosomes would be sterile? Would there be a problem? Yes. Right. You got an odd number. Would it be a problem with tetraploids? No. As long as it's an even number, you're okay. So a lot of the odd numbered polyploids, they seem to, I guess because they grow vegetatively. All right. So there are, I guess, a few examples in animals. We're not going to go into those. All I want you to do is just be aware that polyploids most common in plants, and that they really that you understand the difference between aneuploid, which is a gain or loss of 1 or 2 chromosomes, versus polyploids, which is a gain of entire sets of chromosomes. Okay. Okay, that went well. No, I think on some of those, like the crossing over with the inversions, you really want to sit down and look at that and trace them out and then convince yourself that you really understand what's going on there with the translocations. Now, if you know any other questions on that chapter, or do you have to wait till it's a little clearer in your head to be able to ask questions? Okay. Now we're going to have to change gears here a little bit. We're going to just do, this is the only chapter we're going to talk about bacterial and viral genetics. Now, sometimes, depending on the timing, these topics come up in microbiology, so some students find there's sort of certain level of synergism that they get it at the same time. Unless they get conflicting information, that could be another problem. Okay. So we got to think about things a little differently. These are going to be bacteria. They're haploid. The cell structure is different. They don't have a nucleus. So that right there should just intuitively tell you that things are going to occur a little bit differently. So we're just going to go through the genetics of bacteria and viral systems. Most of the emphasis will be on bacterial systems, and there our main example will be E. coli. So why does anybody know why E. coli really came to the forefront as the sort of de facto bacterium for research purposes? I mean, at one point it was said that E. coli was the most studied organism on the planet. Yeah, but why not, you know, Staph aureus or something? Yes. Yeah, they grow fast. Some strains of E. coli are pathogenic, but most aren't. But a lot of other bacteria grow just as fast. They have plasmids, but a lot of other bacteria do.
It's just because some guy decided to study it.
That's it. Guy named Max Delbruck had just gotten his PhD in physics, and this was about the 1940s or so. The atom bomb, the atom's been split. He decided all the big questions in physics had been answered. He was wrong on that one. And he wanted to study something really complicated, so he thought he would try genetics. And he organized other investigators and basically said, why don't we all work with the same research model organism so we can compare notes. So it's sort of, E. coli is not even a good prototypical bacterium, but it just emerged because of this nucleus of investigators who all decided that, yes, this is the organism we're going to use. And so in the early days, sort of before you could do molecular cloning and such, if you really wanted to do genetics, biochemistry and such, you really had to just work with bacteria and viruses because eukaryotic cells were too complicated and the tools to be able to study them just weren't around yet. So that's what we're going to do in this, digress for just this one chapter. Otherwise, everything else from the rest of the course is all going to be on eukaryotic genetic systems. Okay. So one of the classic ways you started with the genetics, working with these bacterial systems, was to look at auxotrophs. So all the so bacteria, especially, we'll just use E. coli, can synthesize all of the compounds they need, all the nucleic acids, all of the carbohydrates, lipids, DNA, RNA, everything they need from just some basic elements, a carbon source, some phosphate, some nitrogen. And so we can put them on so-called minimal medium, which is just a mixture of salts and a sugar source. And if they are fully wild type, they grow fine. Auxotrophs just simply mean this particular strain has a mutation that now is preventing it from making one of these essential metabolites. So a lot of them were focused on the amino acids, so 20 amino acids. So a leucine auxotroph simply means this strain of E. coli will not grow on minimal medium unless I supplement it with leucine. Right, with leucine. You can do it with histidine, do it with any of them. Finding these mutants was pretty much a brute force process. It was called using something called replica plating. So you had these little round discs, kind of like sponges, or they were actually velvet was what was used on those. The idea was you take a bacterial culture and you're going to go through a process that's called negative selection. What we mean by negative selection is the isolate that you want is going to be the one that doesn't grow. So that's one reason it became this brute force problem, because you're going to have a master plate that has all of the cells and another one, a test cell, because the one that's missing here, you got to go find the viable one over here. So you had to do replica plating. So you had a bacterial culture and you're trying to say, okay, Somewhere in here, there should be some bacteria that can't make leucine. How do I find those? So you spread them out on Petri plates. Everybody's done this. You spread out the bacteria. They grow in little colonies. And you're using this little velvet pad basically as a stamp. So you just gently touch the surface of this plate, and you get a sample of the colonies in the exact pattern that's on this plate. And then you touch it once to a plate that's just minimal medium, and in the second plate, a medium that's been supplemented with leucine. So you're putting down, you might have several hundred bacterial colonies on there, but you're making replicas of those. Then you look and you say, okay, these colonies are growing in the same pattern as this one. However, there's a colony right here that's not growing here. So it's negative selection. The one that failed to grow is the one that you want. And then you come back here and you select that colony and double check it and make sure that in fact it's a leucine auxotroph. Okay. So this is back when, this is probably why they had graduate students, because they're the only ones who will sit here for hours and hours on end doing these kind of brute force experiments. Okay, any questions on how that works? All right. A little bit about the bacterial genomes. As I said, the reason people worked with bacteria before some of the newer technology was available is it's smaller. It's easier to work with. So in most bacteria, the genome is circular. Is the DNA circular in the genomes of eukaryotic cells? No. So there's one difference right there. So in eukaryotic cells, the DNA is divided up into chromosomes, and each of those chromosomes has one single double-stranded linear piece of DNA. Most bacteria have a single circular chromosome. Is it going to be double-stranded DNA? No. They're single-stranded? No, it's all double-stranded. So the only exceptions I know to double-stranded DNA as being the genetic material involve viruses. So these are living cells. So it's double-stranded DNA just like we have in our cells, except it's organized into one chromosome and it's circular. All right, and they're much smaller. So just, you don't have to memorize these exact numbers, but I think you need to get kind of a scale of magnitude difference. So the E. coli genome is about 4.6 million base pairs, MBP, and that makes it about 1/1000th the size of the human genome. So our genome is about 1,000 times larger than the E. coli genome. But we only have about 4 times as many cells, or 4 times as many genes. There's still a lot of DNA packed into there. This is kind of a classic picture for the molecular biologist anyway. This is an E. coli, which has probably been treated with lysozyme to start to form some little pores and little holes in the structure. And basically, this is all the DNA just spewing out of the bacterium. So even for E. coli, there's a massive amount of DNA that has to be packaged into this very small cell. The other thing that the bacteria have, was mentioned earlier, are plasmids. These are not common in eukaryotic cells. There's only a few exceptions. Plasmids, you can think about them, they're just little mini chromosomes. They're small, double-stranded, circular molecules. So they're going to look like the main chromosome. They're just smaller. Depending on the particular plasmid, there may only be 1 or 2 copies per cell, or there could be many copies per cell. Here's a very important point. You do not find essential genes on plasmids. Would you find the gene for DNA polymerase, for example, on the plasmid? No. What you do find on plasmids tend to be transposable elements and antibiotic resistance genes. So it carries genes that may give some advantage to the cell, but they're not actually essential for the cell to grow. just to give you just that general size, so we just said the E. coli chromosome is about 3 million base pairs. That makes it, what, about 3,000 kilobase pairs. In comparison, most plasmids are around 10,000 base pairs. So they're just a small fraction of the size of the main genome. One of the larger ones we'll talk about is the F factor. When we get into the sex life of E. coli, it's around 100 kilobases, but it's still much smaller than the main chromosome. And as I said before, plasmids in eukaryotic cells are very rare. Yeast, which is a single-celled fungus, so it's a lower eukaryote. It does have something called the 2-micron circle. So it is one of the few eukaryotes that has known plasmids, but plasmids are very, very common in bacteria and very, very rare in eukaryotic cells. All right. So let's make it complicated. An episome is a plasmid that has two options. It can either maintain itself independently, as we've been talking about, or it can insert itself into the main chromosome and be part of that main chromosome and turn around and come back out again. So episomes are plasmids that can move in and out of the main chromosome. And this F factor that we're going to talk about in some detail is an example of one of these episomes. So all episomes are plasmids. Would that be true or false? That'd be true. But all plasmids are episomes. False. Okay. All right, so let's try this question if I don't give away the answer first. I'll go to 1 minute. Okay. So what's the answer? There you go. Oh, we agree this time. Good. At least we got some agreement. Okay. So let's talk about the sex life of E. coli. You didn't think they had one. Okay. Fundamentally, yeah, there should be some differences here. So what we're really talking about is a form of sexual reproduction with E. coli. Normally, you think about bacteria, they just divide asexually, and you just basically have a clonal population. They're all genetically the same, except for some random mutations. But what we're talking about here is an actual mechanism called, the one we're going to talk the most about is conjugation, where there's actually a physical exchange of genetic information between two different bacterial cells. Okay. A couple of ways to look at how does this compare to replicate or reproduction in eukaryotic cells. All right. First of all, DNA exchange and cell replication are not coupled in bacteria. All right. When you have meiosis and mitosis in eukaryotic cells, you also, that is coupled to cell division, and you end up with two cells. You're not going to end up with extra cells at the end of this sexual reproduction for the bacteria. DNA that's not recombined into the chromosomes is degraded. And so the main thing here is we're going to see that the genetic exchange is a one-way event. There's going to be a donor cell and a recipient cell. The donor cell is going to transfer genetic information to the recipient cell. It's only going to go in one direction. The donor cell is going to remain exactly genetically the way it was. It's only going to be the recipient cell that was changed. So it's quite different than what the typical sort of meiosis. So you got to think about this in a different way. Here's a cartoon for the 3 most common ways that genetic information, DNA, can be physically exchanged between bacterial cells. The first is conjugation. We're going to talk about that in some detail. The second is transformation. What transformation means is some bacteria naturally, or we could say naturally competent, they can take up DNA from their environment. Our good friend Neisseria gonorrhea can actually do this, okay? So these cells are taking up DNA from other degraded bacteria and such, and they're actually, this DNA is crossing the cell wall, crossing the cell membrane, getting into the cell and recombining into the chromosome. So when we have DNA being brought into the cell, we call this transformation. We'll only talk about this very, very briefly. And transduction means that a bacteriophage or a virus was involved in the process. This was a very important technique in the early days of putting together genetic maps of viruses and eukaryotic. So doing some real fine structure mapping. That's really not done anymore because DNA sequencing is just so fast and so cheap. So I'll just talk about this very briefly just so you mainly know about it. So the first one's the one we're going to go up into some detail on. Okay. So this is as we go through the course, I'm going to have several of these landmark experiments. These were key experiments that led to major understanding in genetics. And one of the reasons for having you go through this is to see the thought process of how you ask the question, design an experiment to answer that question, and then see what the outcome is. In most cases, at least my own experience has been, you have some preconceived idea of what the outcome of the experiment is going to be, and that's not what happens. Bacterial systems don't work that way. Or biological systems. But at least directs you to the next one. In this case, this is the final product. So these are landmark experiments. And if you look at them, they're really not complicated. But they just asked a very basic question, set up a very basic experiment, and came to an answer. So Letterberg and Tatum did this sort of classic experiment that proved this process of conjugation. So this looks a little bit complicated at first because there's 6 different genes involved. But here's the main point. There was a strain of E. coli called Y10. It was a threonine. Threonine is what? A nucleotide? Amino acid, okay. So it's a threonine auxotroph, also leucine auxotroph, that's why the minus, and it had to have thymine. But it was wild type for biotin, phenylalanine, and cysteine. This other strain, 24, just had the complements of all of those. So if you look here, whichever's mutant here is wild type here, and vice versa. So the important point is, there's 6 different genes here, but neither strain could grow on minimal medium. Does that make sense? So they mix them together, let them grow for a while, plated out a sample onto minimal medium, and a few colonies grew. Was that an expected outcome? You would have expected what? Nothing to grow. But in fact, a few colonies grew. Yes?
Isn't that like an example of genetic complementation?
No. Yes. We usually think about the genetic complementation more with diploid eukaryotic cells, but in the sense that one is going to provide the missing genetic information, you could make an argument that it is a form of complementation. Okay, good.
Isn't that more for testing?
Hmm?
Isn't that more for testing?
Right. It's not. This isn't the context in which you would use it. But you again, if you're doing an essay question, you could. Yeah, I can see that in a graduate course. Okay, make the argument for me why this is in fact a form of genetic complementation. But it's not something you normally think about with bacterial cells. But the concept is there because what's going to happen is one strain is going to provide the information, the genetic information that the other one's missing. So the first thing this data tells you is what? genetically, at all 6 of those genes, those cells that are growing on minimal medium must all be wild type now. So somehow there was genetic exchange between these bacteria so that at least a few of those bacteria now had all the genetic information they needed to be able to be, which were now prototrophs. That is, they could grow on the minimal medium. So Bernard Davis extended this observation a little bit. They didn't know what it was. Could be a virus, who knows, it could be some factor the cells secreted. They didn't really know what it was. So he did the same experiment, but he used this U-shaped tube and had a little filter right in the middle of the tube down here, which was porous, so that soluble materials could move back and forth. But the pores were too small for bacteria to cross. So he put one strain, he put strain A over here, strain B over here, and what did he find? No growth. So what does this tell you? There has to be physical contact between the bacteria in order for this process to happen. Okay. Pretty straightforward experiment. Okay. Why did I put this up here? Oh, I just gave you the answer. What the hell? We'll see who's awake. I should put that at the end and see who can. Okay, how many of you can remember what I said 30 seconds ago? They're good. How many of you can remember this on October 7th? That's another question. Okay. Short-term memory loss is a bitch. Huh? I've done it before. I put iClicker questions on there. But then I rewrite them. So wouldn't even have to redo it. All right, we'll go to 1 minute on this one. Yes? I'll show you, it's only a single strand. That's why it doesn't lose information. The cell will repair it and make it double-stranded again. I'll show you. Yeah, it doesn't actually lose it. All right, what's the answer? There you go. All right. So here's what happens. This process of conjugation is literally a physical transfer of DNA from one cell to the other. So we're going to have the F minus. So the F minus cell is going to be the recipient cell, and the F plus is going to be the donor. The F plus cell is the cell that has this episome, this plasmid, called the F factor or fertility factor. Among the genes on that plasmid are the proteins necessary to make these fimbriae. They basically create this pilus. This is a hollow protein tube that literally connects the donor cell, the F plus cell, to the recipient cell, and there's going to be a physical transfer of DNA from one cell to the other. Now here's the key point, and it should show up in the next slide or two. Only one strand of the double-stranded DNA is going to get transferred. So, since DNA is complementary, the DNA repair system in the donor cell can repair that and make it double-stranded again. So the donor cell is not going to lose any genetic information. So the donor cell is going to remain genetically identical to where it started, but new genetic information is being transferred in to the recipient cell. Okay. So this DNA transfer is always one way, from the F plus to F minus, and it always starts at the same point. So there's a DNA sequence on that F factor called ORI-T for the origin of transfer. The DNA becomes nicked. This is a jargon term. It means one of the phosphodiester bonds between two adjacent nucleotides gets broken, but only on one strand. So the circular DNA remains together, but you've broken the covalent bonds between the DNA strand, and you can literally just sort of peel that DNA off. Because what's really holding the two strands together? The covalent bonds holding it together? Well, it's just hydrogen bonding. So the bond forces are much, much lower. Okay. So what's going to happen is the single strand of that F factor is going to get transferred. If the entire F factor gets transferred, and most of the time it does because it's very small, the F minus cell now becomes F plus. So we're going to start out with an F plus and an F minus, and we're going to end up with two F plus cells. Okay? So here's the cartoon of it. This is the main chromosome. Here's the F factor. These are not drawn to scale. All right. Here's the pilus. Here's the key point, is just one strand is being transferred. And it always starts, so this position right here, this DNA sequence is designated as ORI-T, because this is where the nick occurs on one strand. And literally this just rolls over, and it's getting, so the single strand of DNA is getting transferred into this recipient cell. Meanwhile, the DNA repair system is making this double-stranded again. Same thing's happening over here. This is becoming double-stranded. And now you have two F plus cells. So in the traditional sense, the F plus cell would be considered the male cell for E. coli. This is a female. And so we have kind of a Bruce Jenner effect here. So it's one way to help you think about it, right? Okay. So E. coli can actually undergo a sex change at a much lower cost. Okay. It's part of its natural life cycle. Okay. Now, in the next few minutes, I want to really complicate things. So stay with me. Because does this explain, okay, here's a very important point. I'm giving you all these pointers as to how to get these questions right on the exam, okay? Are there any essential genes on this F factor? No. Are the genes that are encoded, that were required for those auxotrophs we were talking about, threonine, biotin metabolism and such, would those genes be on the plasmid? No, those would be on the chromosome. So how did, in fact, genetic information from one strain to the other that should have been part of the chromosome, would it have gotten transferred in this way? It shouldn't be on the F plasmid, right? So what Letterberg and Tatum were looking at, they didn't realize it at first, was the F factor is this episome. So what they were working with is what was called an HFR strain. It gets its name for high frequency of recombination. All that means is occasionally, about 1 in 10,000 cells that are F plus, that episome integrates into the chromosome, and it's now just part of the main chromosome. When the F factor is separate by itself, that's an F plus cell, and then it can make an F minus cell an F plus. Or that F factor can integrate into the main chromosome, and when it's part of the main chromosome and it replicates with the main chromosome, we call it an HFR, for high frequency of recombination. All right. Here is the important point and why it could be used for genetically mapping the E. coli chromosome. The F factors, you can still undergo conjugation exactly as you did when it was just its own separate plasmid. But now it's going to drag some of the chromosomal DNA with it. It's still going to start at ORI-T. So the important point here is we're going to have different strains of E. coli that have this F factor integrated in different places. But the transfer from the HFR to an F minus cell always starts at the same place. Okay? So now remember, the E. coli chromosome is much larger than that F factor. So transferring that F factor from an F plus cell to an F minus is very efficient, probably takes a minute or less. Transferring the entire E. coli chromosome, all 3.4 or so million base pairs, takes about 100 minutes. So the E. coli genetic map is mapped out in minutes based on how long it takes to transfer from one gene to the next. It's all based on these conjugation studies. So the time that it takes to transfer the genes gives you the relative distance between the genes. So let me go through this real quick, and then we'll use this as a point to start again next Monday. So the idea is that we're going to conjugate, we're going to make these HFR strains with an F minus strain, and then stop the conjugation. Basically, these experiments were done about the time that Waring invented the blender. So basically, the blender was used to generate enough shear forces to break the pili and such. I'm not sure you want it back for your bar when they're done with it, okay? But it was some new technology at the time. So this is a kind of a busy-looking slide, but the whole idea is wherever the F factor integrates into the chromosome, that's where the transfer begins. And then the genes start transferring over, and they just literally are pulling the chromosome sequence. Now again, it's just a single strand. So the donor cell is going to remain genetically the same. How much genetic information gets transferred depends on how long the conjugation goes on. Most of the time, you're not going to get the whole chromosome transferred because it's a very delicate union between these two cells, this little tiny protein bridge, and it's got to last for more than an hour and a half to be able to transfer all of the genes. But all we're really doing is just measuring how long it takes for these genes to transfer over. And so if you look at this graph, what you begin to see is you can just sort of plot out how long it takes for the recipient cells, which will be the mutants, how long it takes for them to get the wild type copy of the gene. Let's see. So if we look at this data, what is this really telling you? In this case, which gene is transferring first? Okay, so on the y-axis, we've got the percentage of cells that have a particular trait. So which one goes fastest? You get azide resistance first, then you keep, as you kind of move down the list, and that's what they're trying to show you over here. Now, as I'm going to just do this real quick because we can come back, so you can think about it, and we'll come back on Monday. So this is going to be unidirectional transfer. Now, here's the important point. The F factor in different cells can integrate in different places. But it always begins transfer from where it starts. So here's a strain just called HFR1. This is just one strain. So here's the F factor. The arrow indicates the direction of the transfer. To make it more complicated, depending on which way the episome integrates, it either is going to go clockwise around that circle or it's going to go counterclockwise. The direction of the arrow indicates the direction. Okay? So leucine is going to transfer first, then threonine, thymine, histidine, etc. Okay? Could you map out the chromosome if that's all the data you had? No, you need it starting at other points. So this is HFR1. Well, here's HFR5. Well, it just integrated at another place. This time it's going to be thymine, threonine, leucine. So if you line these all up, here's 6 different strains. Do you see how you put the genetic map together? Well, do this. Move them around, and what you see is they all line up. They're just starting the transfer at different points. but the order of the genes is still the same. And so by putting all of these together and lining them up, you can see that this is the order of the genes, and the number of minutes that it takes for the first one to appear and then the next one gives you the relative distance between them. Okay? So let's just, we'll end this with this simple question. Let's do this one. I think I may have a second one on there, and then we'll call it a day. As I said, I know I went through that very quickly. Let's just let you see it. We'll come back and we'll do this again on Monday. Important thing to remember here when it comes exam time is you don't have these essential genes are not on the F plasmid. So you have to be working with an HFR. Okay. Okay. Any other questions? All right. It looks like. Okay. So we'll start again and review this on Monday. Otherwise, have a good weekend. Say what? Oh.